Hello, everyone. I'm Jingtao Wang from McGill University. And my project is called Enabling Affordable Single-Cell Data in Large Cohort Studies via Deep Generative Neural Networks and Active Learning. And here's the overview of my presentation. We'll start with motivation, then method. We develop a tool called SC Summer Profiler, and then results on publicly available data sets, and then discussion and acknowledgement. We start with motivation. So traditional bulk gene expression data, or it could be RNA sequencing data, are cheap, but they only provide average expression of a group of cells. On the other hand, more advanced single-cell gene expression data can provide more higher resolution information. They can provide gene expression data in individual cells, but they are much more expensive. So we're trying to utilize the cheap bulk data to in silico generate single cell data so that biological study can have affordable single cell sequencing data. To do that, we developed a tool called a single cell Summer Profiler, and we proposed a concept called Summer Profiling, which means for a cohort that you're interested in, you want to study, instead of getting the single cell profiling for all the samples in this cohort, we get the cheap bulk data for all of them and only get the single cell data for some representative samples. And with these two types of input, we output the single cell for all samples, either real profiled or in silico generated. So let's go over the pipeline of our method. So given our interested cohort, we start with the initial setup step, which includes bulk sequencing for all samples, and perform a clustering analysis for the bulk data. Then we select some representative samples. Those samples are the samples close to the cluster centroids. And then for these selected representative samples, we perform single cell sequencing to get the single cell data and do some processing, which includes basic preprocessing and some feature augmentations. And then for all the other samples, we call target samples without real single cell data, we use a deep generative learning model to generate single cell data for them. So each time for a target sample, we input the target sample and its corresponding representative samples, bulk data, and the single cell data of the representative sample. Then this deep generative learning model first to try to reconstruct the single cell data of the representative. Then the bulk data's information is going to guide the model to alter this reconstruction process to inference process to infer the single cell data of the target sample. Once this is done, all these samples will have single cell data, either real profile or in silico generated. And if budget allows, we use an active learning algorithm to select the more representative samples to enhance the Summer profiling performance. This is done by first identify the most heterogeneous sample cluster. Then we find an optimal split of this cluster to minimize the heterogeneity. Then we do a clustering membership update. And after this iterative process, we end up with a single cell cohort. Then all the sample will have single cell data. We can do all kinds of single cell level downstream analysis. Then let's take a closer look at this inference step. So how exactly do we infer the single cell data for the target sample? This is the most important component of our pipeline. Again, the input each time is a representative sample single cell data and the bulk data of the representative sample and the target sample. And we're trying to output the target sample single cell data. And three main steps: reconstruction, then introduce bulk information to alter this reconstruction, and then generate single cell for the target sample. And here are some visualization to help people better understand what exactly it means. So let's say the blue cells are the representative samples cells distribution. And what we want to get, the goal of the inference is getting these red cells, which are target samples, ground truth cells. So there are two different samples. They have different cell distributions. We do this by first reconstruct the blue cells. Yellow cells means the computationally generated cells. And we push the yellow cells towards the direction of the red cells. And if in the end, the yellow cells are very similar to the red cells, then we say the in silico inference of the red ground truth cells are successful. And we also show some real-world example generated using our method. This is the reconstruction. Blue cells are the representative sample cells, and those gray cells are the reconstruction. And they are quite similar, meaning that the reconstruction performance is good. Then move forward to the inference step. We move the generated cells towards the direction of the ground truth. Here we can see that the yellow cells are actually more similar to the red ground truth cells than to the blue representative cells. It means that we can actually infer the red ground truth cells very accurately. And we'll get into the method, how exactly we achieved that, how we did that. So we reconstructed the representative single cell data with a variational autoencoder. Then we add a discriminator network to this VAE, making the overall structure a VAE GAN to enhance the reconstruction performance. Actually, only with VAE, it's almost impossible to get the perfect reconstruction. So this discriminator is really important for enhancing the reconstruction performance. And then we add one more constraint to this VAE GAN model. We require the generated single cell data have average values similar to the bulk expression of the representative. This is because by definition, bulk is the average of single cell data. And this step is important because it gets the model ready for the next inference step. Then up to now, it's still reconstruction, but in the inference step, we substitute this bulk gene expression of the representative with the bulk expression of the target sample. So now we require the model to generate single cell data with average values similar to the bulk data of the target sample. So that's how we introduce information about target sample to this reconstruction process and gradually guide the model to generate single cell data for the target sample. Here's a visualization of the whole process. So first, we do reconstruction. The yellow cells have overlap with the blue cells. It's still reconstruction of the blue cells, representative cells. It's still very different from the red cells. And the second reconstruction stage is still very different from the red cells. Then we start to move those yellow cells in the inference stages towards the direction of the red cells. We gradually move them then until in the end, after the final inference stage, we can see that the yellow cells are actually more similar to the red cells than to the blue cells. So this is the whole process of the inference. Now we show our results on publicly available data sets. We first evaluate our methods using a COVID-19 data set with 124 samples with single cell data. We generate pseudobulk data for all the samples and select 28 samples as representative samples. And we run our pipeline to generate a semi-profiled cohort and compare the similarity with the real profiled cohort. In the UMAP visualization, we can see that the panel A is a real profiled cohort, and panel B is the semi-profiled cohort. They are quite similar. Different color represents different cell types. And C is plotting two data sets together. They have a lot of overlap, means the two cohorts are quite similar. And then we also plotted the semi-profiled cohort using different colors to represent the representative cells and the in silico generated cells. And we can see that actually more cells are generated by the deep generative learning model. Then we also show that our method also works well as a deconvolution method, which is estimating the cell type proportion in bulk samples. In the first panel, we accurately estimate the cell type proportion in each disease conditions. And in the other panels, we compare with the existing deconvolution methods. The first four are our methods with different settings. They show much better results than existing methods in terms of Pearson correlation, CCC, and RMSE. Then we also show that if we evaluate the similarity using gene expression values, the two data sets are also similar. The real profile cohort and semi-profile cohort are similar, have similar interferon pathway activation patterns. And in the panel on the right-hand side, we show as more samples are selected as representative samples, we can gradually decrease the error quantified using gene expression value. We also compared with a baseline method called selection-only method, which is a higher curve here. For the selection-only method, we always have the same representative samples, but whenever we want to infer the single-cell data for our target sample, we simply copy its corresponding representative. So it's doing naive in silico inference. And it shows that our method using the deep generative learning model actually has a much lower error. And for more in-depth analysis, like biomarker discovery and enrichment analysis, we also show the semi-profile cohort can actually result in have very similar results with the real profile cohort. So here is the biomarker expression pattern, and this is our HO plot quantifying the similarity of the biomarker. Brighter color means higher similarity. And we also show the enrichment analysis are also quite similar. And if we do more in-depth analysis like cell-cell interaction analysis and the pseudotime analysis, and they also show the semi-profile cohort and real profile cohort are also quite similar. We also do all these analysis using a more heterogeneous and challenging colorectal cancer data set, and we also show the results are quite good. And then, but for the COVID-19 data sets, all the bulk data are actually pseudo-bulk data generated using single cell. But that's quite different from the real world setting, because if people want to use this in real world, they have real bulk data coming out of sequencing machine, which is quite different from pseudo-bulk data by averaging single cell data. This is because the single cell data is much more noisy than bulk data. They have a lot of dropouts, for example. So we want to test our method, see if it still works on data sets with real bulk data. So we find this data sets with both single cell and bulk data for all the samples. And we do the same analysis and show the semi-profile cohort is still very similar to the real profile cohort. And this is also true for all the downstream analysis. So to conclude, we develop a deep generative learning model that can perform in silico single cell inference for a target sample based on the bulk data and a single cell reference. And we also design an active learning algorithm that selects the most informative representative samples to maximize the in silico single cell inference performance. And we believe the impact is that we can provide a more affordable single cell alternative single cell data for any single cell studies. And here are the links to our manuscript, which is accepted by Nature Communications and our GitHub repository. Or you can find our tool by simply searching SC Semi-Profiler. If you want to use our tool in your study, I will be happy to help. Please let me know. And finally, I would like to thank everyone from the 2 Lab, Dean Lab, and Fonseca Lab at McGill, and our fundings. And that's the end of my presentation. I'm happy to answer any questions. Thank you.
Thank you, Jingtao. So do we have questions? I'll definitely get yours.
Hello, can you please explain a bit how did you make the cell graph in the deep generative model,
Oh.
the pipeline?
I think you noticed a detail that I went very briefly. Do you need the cell graph?
The cell
Oh,
graph on top. How
Yeah.
do you construct it?
Good catch. So these cell graphs are generated. This cell graph using sklearn's functionality. It's just a KN graph. It's cells with similar gene expression will have an edge, and then to utilize that in the model, so we use the graph convolutional layer to aggregate information from neighbor cells to enhance the model's performance.
What was the similarity metric that you used?
Similarity is just based on a gene expression value.
No, did you use cosine similarity or something else?
I believe sklearn uses Euclidean distance similarity.
Okay. I get it. Thank you.
No problem.
All right, nice talk. So you said that the generated single cell is an average. You set it so that it's an average of the bulk, but sometimes the bulk. I mean, the single cell. When you do the isolation single cell, you get different amounts of the different cell types. This is, if you're doing PBMCs, it's pretty evenly distributed. But if you're doing solid tissues, one cell type might not isolate well, and so they might not look like the bulk data might not average out to the single cell. So might it be better to use a weighted matrix? So when you're doing the training, so that it learns the single cell distribution that you're isolating, and then use that as for learning. Do you understand what I'm saying?
Do you mean some gene features might be more important in some cases?
No, just that, if you're, so for example, if you're isolating liver samples, the PBMs, the immune cells are readily come out in single cell, but the hepatocytes, you don't get much data from that. So you get very few hepatocytes in that. But if you just do the bulk isolation, you're going to get a strong signal from hepatocytes. But if you do the single cell, then you only get a few hepatocytes, and the immune signature will be much stronger in that. So,
I think I understand your question. So let's think about this. I have a representative sample and a target sample. If single cell sequencing is always going to miss that specific cell type, then for both, for the single cell data for both samples, we'll have a fewer of that cells of that cell types, right? That's true, right? And maybe for the bulk data of the both samples, then we won't capture that signal. Actually, in our method, the model only deal with pseudo bulk. So even for the target sample, which we do not have the single cell data, we also estimate the pseudo bulk data for the target sample. So we try to use this to avoid any technical difference between the single cell data and single cell data and bulk data. So in details, it's like this. For example, I have both samples have bulk data, and I compute a fold change between these two bulk data, and I will have pseudo bulk for the representative sample, and I use this fold change and a pseudo bulk of the target sample, a pseudo bulk of the representative sample to estimate the pseudo bulk of the target sample. So the model only deal with pseudo bulk. So this is a way that we bypass any technical difference between the bulk data and single cell data. We have more details in the paper.
Yeah, so very nice talk. Two questions. One, how sensitive is this? So did you look at rare cell types and how well you can reconstruct? Because that's always the challenge if you're very, very rare. And the second question is about this representative sample. Why do you use one representative sample? Why not use everything with some weights or at least a few? Because it seems very dangerous to rely on one sample to reconstruct where you can have a few that are pretty similar. Why just use one for the reconstruction?
So for the first question, we didn't specifically look into the deconvolution accuracy of the rare cell type, but on the UMAP, we checked this UMAP. It seems that even for those rare cell types, the reconstruction of the in the two versions, they're still quite similar. The clusters have almost the same cell types, same shapes, and the same amount of cells. But we didn't check in the deconvolution performance using some kind of metrics. That's something we should look into in the future. And for the second part, that's a very nice question, because that's actually a future direction. We indeed think for the representative and target sample, if the cell type proportion are too different, it's hard for the deep generative learning model to change this. So a better idea would be use a combination of multiple representative samples, and if we have a prior of a cell type proportion, and we sample cells from multiple representatives, that would be a better idea, and actually a future direction. Yeah, and yeah, thank you.
Okay, I hear